SiriusXT has developed a laboratory-scale soft X-ray imaging microscope capable of imaging the whole internal structure of intact biological cells at high resolution.
Yeah, X-ray microscopy enables 3D imaging at ultrastructural resolution of intact cells in their near native state without chemical fixation. So this permits facile detection of subcellular changes that can arise in various disease states, leading to a much deeper understanding of diseases such as cancer or Alzheimer's.
Despite the billions of euros of investment in science and cell biology over the years, we've barely scratched the surface in our understanding of so many human diseases. I think at this point in time there's a clear and present need for innovative imaging approaches and technologies that can understand human diseases in the context of intact human cells.
Cell shape and the shape of its internal organelle are seen as important indicators of the DNA mutations that take place in the genes of cancerous cells as they split and multiply uncontrollably as the disease spreads. For this reason, 3D imaging of the internal structure of whole, intact cells is playing an increasingly important role in helping scientists to understand cancer.
The only technology available today that can image through a whole cell without needing to slice it or stain it is soft X-ray imaging.
The problem is that the soft X-ray illumination required for a soft X-ray microscope is only available from electron accelerator facilities called synchrotrons. Only 4 of these billion-dollar football stadium-sized facilities exist that have a soft X-ray imaging beamline, and only a small number of cell biologists have been able to get access, having had to queue for up to 12 months to do so. Despite this limited access, numerous breakthroughs in understanding disease causation and diffusion have been made using soft X-ray imaging, both as a standalone imaging modality as well in correlating soft X-ray images with super-resolution fluorescence images. SiriusXT has invented a means to miniaturize the synchrotrons into a small chamber that will easily fit on a laboratory bench. This chamber produces a continuous focus beam of soft X-rays that can be used to illuminate the microscope and to image through the full cell, just like with a synchrotron beamline. This patented innovation will give cell biologists access to soft X-ray imaging in their own labs, opening up the benefits of this imaging modality to thousands of cell biologists working in disease research.
The ability to image an entire cell in 3D enables detection of rare events within the cell. This property is extremely important and makes X-ray microscopy unique. For example, in drug delivery studies, it reveals key subcellular changes which can easily be missed by conventional electron microscopy, where only a few sections from the entire cell are actually examined.
In an imaging core facility, we need a correlation between all the imaging modalities, and soft X-ray is a perfect match to increase our capabilities to provide the best service to our users.